Hello and good evening, everyone, depending on where you are in the world. It's at the top of the hour, so we wanted to get started and welcome all of you to the third in our webinar series that focuses on the state of the science related to gut microbiome, diet, and health. My name is Marie Latalipe, and I work with the Gut Microbiome Committee of ILSI North America that in part organized today's webinar. And for this series, we had the opportunity to collaborate with the American Society for Nutrition, and we're extending this out to the membership of ASN, and I think many of you have joined through that venue. A couple of housekeeping items. The webinar is recorded and it will be available on our website as a resource, so you can go back and listen to any components of particular interest to you. And at the end, we'll open this up for questions. At the moment, everyone is and will remain on mute, but you have the opportunity to type your questions into the question box there, and we will be reading the questions for the presenter. And with that, I would like to turn the microphone over to Dr. Wendelin Jones, the Executive Director of ILSI North America, for a few opening comments.
Great. Thanks, Marie, and thanks to all of you for taking the time out of your day. For many of you, we know it is evening, so thank you for joining us perhaps immediately after dinner. We are thrilled that we've got so many participants joining in on this webinar. For those of you who are less familiar, ILSI North America is a nonprofit scientific organization that advances food safety and nutrition science for the benefit of public health. We do not lobby or develop policy positions. Rather, we are focused purely on the science. We do our science through our unique operating model of bringing together folks from government, industry, and academia. That tripartite model allows us to catalyze science for credible decision making. One of the things, and you'll see this on the bottom right hand of the slide, that we're particularly proud of is our platinum seal of transparency from GuideStar. This is a certification that we are awarded because not only do we conduct our work in a transparent manner, but we are transparent about our progress against our goals, delivering against metrics, and ensuring that there is that robust aspect of sharing our information. So we wanted to highlight our platinum seal of transparency and note that we're one of a very small percentage of nonprofits who have achieved that. As we transition to the next slide, I just wanted to quickly note, if you're interested in learning more about ILSI North America, please visit our website at ilsina.org. You can follow us on Twitter at @ILSI_NA, or of course, follow us on LinkedIn. It's through our website as well as our social media engagements that we look to support and advance the science. So want to thank all of you for your time. Want to thank our collaboration with American Society of Nutrition, and of course, thank our presenter. With that, Marie, I'll turn it over to you.
Great, thank you, Wendelin. And I just made Dr. Berean the presenter. So I'm pleased to introduce to you Dr. Kyle Berean of the Royal Melbourne Institute of Technology and the University of New South Wales, as well as Atmo Biosciences. Dr. Berean joins us in the morning, his time, tomorrow, so thanks for getting up so early. Dr. Berean is a co-inventor of the Atmo gas sensing capsule, and he's actually co-founder of Atmo Biosciences. Kyle is currently the VP of Technology for the company. He also holds a senior research fellow position at both RMIT University and the University of New South Wales. Kyle has a PhD in materials science and electronics from RMIT, and he currently holds 3 international patents. He's also an author on over 35 peer-reviewed journal publications. So we greatly appreciate his taking the time to share some of his work today on better understanding the role and detection of intestinal gases. And with that, I will turn the platform over to you, Dr. Berean.
Thank you very much. Thanks a lot, Mary. Thanks a lot, Wendelin. And thanks, ILSI, for putting on this webinar. I'd like to thank everybody for attending, and I hope you're all staying safe in these weird times that we live in. So as Mary pointed out, my name is Dr. Kyle Berean. I'm a founder and VP of Technology of Atmo Biosciences. I'm co-inventor of the Atmo gas sensing capsule. To put this presentation into some context, as was stated, is that I have a science and engineering background. I'm not a gastroenterologist, nor am I a dietitian, but I am going to be talking to you about the intestinal gases, the influence that they have on gut disorders, and the role of monitoring their production. So the first reports of intestinal gas measurements do date back to 1781, when Benjamin Franklin discussed the measurement of flatulence and the influence of food on its odor. More recently, however, interest has been growing in the role of intraluminal environment of the gut as an inducer and/or modulator of human health and disease, which has been predominantly driven by an improved understanding of the crucial role of the gut microbiota. However, insight into the functional activity of the intraluminal microorganisms and how they might interact with the host in the specific ecosystem of the gut is still somewhat limited. Displayed here is just an overview of the relationship between the transit of food, specifically carbohydrates, fats, and proteins, which are the main dietary macronutrients. Most digestion occurs in the small intestine, with the absorption of key products of digestive processes for which digestive enzymes are largely responsible. The food components that are not digested pass into the large intestine, where many are digested by microbial metabolic processes. Fermentation is shown in the dark green box. Fermentation is high in the proximal colon due to the plentiful amounts of substrates, but decreases distally, predominantly due to the exhaustion of these substrates. Obtaining an overall picture regarding the physiology of intestinal gases, whether they are produced by microbial fermentation or chemical activity, is the first step towards understanding their influence on human health. The predominant intestinal gases are nitrogen, oxygen, carbon dioxide, hydrogen, and methane, whereas other microbial-produced gases such as hydrogen sulfide, nitric oxide, and sulfur-containing compounds are believed to exist just in trace concentrations. Gases might enter the gut by being swallowed or may be produced via internal chemical reactions or through bacterial fermentation. In the gut, oxygen and nitrogen are almost solely derived from swallowed air. In the proximal small bowel, bicarbonate, which is secreted by the pancreas, chemically reacts with the hydrochloric acid leaving the stomach, and this reaction creates carbon dioxide. Towards the distal part of the small intestine and into the colon, intestinal gases are produced predominantly through microbiota fermentation. The gases hydrogen and carbon dioxide are released during the fermentation of carbohydrates, whereas methane is produced through the metabolism of carbon dioxide and hydrogen by archaea in the colon. Hydrogen sulfide and sulfur-containing trace gases are produced during the fermentation of these proteins and by sulfur-reducing bacteria that reduces sulfates and sulfites. There are large differences in the patterns of gas production that have been noted between individuals, and this has been found to relate both to the nature of the gut microbiota and to the dietary substrates available. Gases produced along the gut are either absorbed via the mucosa to enter the blood circulation or transformed into chemical components via microbial activities. The non-absorbed gases are pushed forward from one segment of the gut into the next, and then finally released as flatus. Interpreting the precise relationship between gastrointestinal symptoms and intestinal gases is the key for generating future libraries that define gas profile signatures, which could be used for prevention, diagnosis, and monitoring of gastrointestinal disorders. It has been shown that the microbiota and gaseous molecules can have important roles in the pathogenesis of gastrointestinal inflammatory and functional diseases. For the motility applications, oxygen can be used as a location marker as the gut becomes increasingly anaerobic throughout its length, with stepwise decreases in oxygen concentration in concert with the increasing abundance in luminal microorganisms. As a result, oxygen concentrations in the colon become very low. Methane has a physiological effect as a gasotransmitter. It has been shown to slow motility in the colon and has been associated with constipation. Bacterial fermentation is the major source of hydrogen and carbon dioxide production in the gut. Owing to its specificity to the gut and the presence of in exhaled breath, hydrogen is the main gas marker of carbohydrate fermentation used for diagnostic testing of carbohydrate malabsorption or small intestinal bacterial overgrowth. Analysis of breath and fecal samples have shown the feasibility of using gut gases and volatile organic compounds in the diagnosis of IBD and colorectal cancer, but this approach is not ready for routine clinical application. Although the assessment of hydrogen and methane gases of the gut generally have minor value in the diagnosis and monitoring of IBD, hydrogen sulfide and volatile organic compounds have demonstrated more success. Indeed, breath levels of hydrogen sulfide and volatile organic compounds have been shown to be elevated in patients with IBD relative to healthy control individuals. Diet has been shown to have pivotal roles in altering volatile organic compound profiles in the feces of patients with IBD. Additionally, hydrogen sulfide has been implicated in the modulation of colonic compliance. For colorectal cancer, an effective gas-based screening approach is still lacking. However, both exhaled and fecal volatile organic compounds have been successfully linked to colorectal cancer, as tumor growth involves several metabolic changes related to volatile organic compound production. Despite the examples presented here, certain disagreements in the field remain regarding whether the gut gases are the cause, consequence, or both of symptoms of gastrointestinal disorders. Current methods of measuring gases in the gut are somewhat lacking. In vitro gas simulators can predict timing of fermentation and can assess the correlations among changes in short-chain fatty acids, microbiota, and gas contents. Such information is useful in understanding the fermentation behavior of colonic microbiota, the effects of diet on probiotics, and the influence of enzymes and dietary interventions on gut homeostasis. Nevertheless, crucial deficiencies of such simulators is the control of mucosal absorption and the luminal transit, and the lack of microbiota diversity. The schematic diagram shown here is the simulator of the human intestinal microbial ecosystem, or SHIME model. Indirect measurement techniques are also available, such as body colorimetry. Where the individual is kept in a gas-sealed environment, the total temperature and gases, in particular carbon dioxide, are measured to assess the overall metabolism. Body colorimetry is frequently used for assessing physical performance and, to a lesser extent, dietary ingestion, IBS, and malabsorption, but it's expensive to set up and the participant needs to spend excessive periods of time locked within a sealed chamber. The most commonly used technique to measure gut gases is breath testing. Breath testing is an indirect measurement process of gases that are generated in the gut, absorbed via the gut mucosa, entering the blood circulation, released into the lungs, and finally exhaled. Generally, exhaled samples are collected in bags, which are later fed into an analyzer. Examples of positive and negative results for hydrogen breath testing in response to carbohydrate loads are shown here. In response to a glucose, lactose, or fructose load, positive findings, indicated by a rise in breath hydrogen concentrations, as seen on the right, might aid in the diagnosis of small intestinal bacterial overgrowth, lactose malabsorption, or fructose malabsorption, respectively. A limitation of the use of breath hydrogen tests is the lack of an agreed standardized procedure. Protocols differ regarding the dose of carbohydrate used, the breath sample interval, the duration of test, the cutoff level for defining a positive test, and the value, duration, and method of defining symptom induction. But what if things were a little different? Imagine if assessing the gases generated in the gut was as simple as taking a pill. We are aiming to become the common method to accurately measure the gut gases at their point of production, with the aim to eventually become a gold standard for baseline screening of common GI symptoms, resulting in targeted treatment, earlier relief of symptoms, and finally savings to the healthcare system. Our system measures the gases within transit of the GI tract, wirelessly sends the data to a receiver attached to a phone, which transmits the data to the cloud for aggregation and analytics by researchers and clinicians. The Atmo gas capsule currently measures hydrogen, motility through an oxygen proxy, and temperature. There is ongoing work to increase the suite of analytes that are measured to include carbon dioxide, methane, hydrogen sulfide, and some short-chain fatty acids such as acetate, butyrate, and propionate. But all of this work is preliminary and still ongoing. During the transit, we use oxygen as our location marker so we can understand where the gases are being generated within the GI tract. From the limited literature on the intestinal gas composition of the entire GI tract, we see stepwise changes in the pressure or the partial pressure or concentrations of oxygen along the length of the gut. The gut becomes increasingly anaerobic with presumed stepwise changes in oxygen with an increasing abundance of luminal microorganisms, resulting in a low oxygen concentration in the colon. As we can see, the motility output from the Atmo capsule, which is an oxygen proxy, it tracks what is expected from the literature quite precisely. Currently, we have validated this marker with only an ultrasound on one patient. We are currently expanding this study to include more participants to further add confidence to this measure. An evaluation for using the Atmo gas capsule in the assessment of motility and transit time has been performed by comparing the outputs to the commercially available product used clinically, the Smart Pill. The Smart Pill here on the left measures pH, pressure, and temperature. Its report outputs gastric emptying time, small bowel transit time, large bowel transit time, and total transit time. These are the measures in which we are comparing, and that is a picture of our capsule on the right. The Smart Pill location shown in the chart in red is the pH output, and its corresponding organ is shown in the red bar on the top of the chart. The Atmo location marker is shown in cyan, with the corresponding organ shown in cyan at the top, in the cyan bar at the top of the chart. The Atmo temperature is shown in purple, and the hydrogen as measured by the Atmo gas capsule is shown in dark blue. This example highlights the similarity in the transit metrics that are delivered, but the contrasting outputs of the two devices. The pH measurement in red is a noisy signal that is very clear when it leaves the stomach into the small bowel through a large pH change from around 1 into neutral around 7 or 8, but it's less obvious in its transit from the small bowel into the large bowel with that subtle 1 pH unit drop. The Atmo location marker does not suffer from such noise within the signal, but the gastric emptying marker is more difficult to see than the smart pill. The transit through the ileocecal valve into the large bowel is very obvious, and in this healthy participant example shows significant hydrogen production once entering the colon. The Atmo gas capsule has also been benchmarked in a simultaneous study against hydrogen breath test, where a fermentable carbohydrate, inulin in this example, in the top, was ingested and compared to an absorbable carbohydrate, glucose, in the bottom. The breath test on the right and the gas capsule on the left are in general agreement for the amount of hydrogen production in both participant arms. The breath hydrogen has a noisy signal and a variable background, as seen in the bottom right. The capsule, however, shows a very high signal-to-noise ratio, as it is measuring percent or parts per 100, where the breath test is measuring parts per million. That's several orders of magnitude higher concentrations. The key here is that the capsule is measuring at the point of production with information on the organ of gas production. And the breath test is indirect with little to no understanding of where the gas is generated. Some of the key benefits of using the Atmo gas capsule are around the measuring of the valuable gaseous biomarkers at the point of production, leading to greater accuracy than breath tests. The data is aggregated in the cloud, allowing for easy access for researchers and clinicians, and all at a very relatively low cost. It's non-invasive, allowing the patients to go about their daily lives after swallowing the capsule that is just a similar size to a common vitamin pill. The data is collected in real time, allowing for plenty of future research to be undertaken to expand the current understanding on the role that gases in the gut have on diagnosis, intervention, and management of disorders and diseases. So thank you very much, and I hope that you've seen the value in measuring gases of the gut and the potential of this exciting new technology. Thank you, guys.